Okay, thank you very much, Maura. Maybe you can stay here on the podium, and I ask the other two presenters as well to share the 3 microphones so that we can do the Q&A.
Let me start the quiz.
Yeah, okay, go ahead.
Okay.
So John will first do a quiz, and then we'll...
Okay, so we got a
Come back to the
quiz coming
questions.
up, guys. You had to pay attention here. All right, so there will be the quiz available. It'll be on both the platform and the app, so it's electronically responded to. This poll is being sponsored by our silver sponsor, Janssen. However, I do need to point out that this poll in no way it represents an endorsement by ICE-HBV or the HBV meeting into any Janssen product, although they were very kind to generate money. And so what I need to do now is hit a green button, and there we go. And so the question is what would you like to achieve with a novel therapy for chronic hepatitis B? This is a multiple choice here, and you're not graded on a curve. And so it's functional cure, which is undetectable HBV DNA and undetectable HBsAg sustained off treatment in at least 30% of your patients treated with a novel therapy. Another one is a partial cure, which is undetectable HBV DNA and low HBsAg sustained off treatment in at least 60% of your patients, and intensified viral suppression in greater than or equal to 90% of your patients treated with a novel therapy. And so that poll is now online, and we will proceed to the question and answer period here. And Fabian is here with us, and he may be able to help us.
Just as a remark, we have until 10:30, right? Because Nora Turo is unfortunately not able to give her talk, so we have a little bit of extra time, and then we have a 20-minute break afterwards. Fabienne, do you have access to the online questions?
I have trouble, so I'm trying to get connected again. I mean,
Okay.
Don't worry about it, Fabian.
Let me start otherwise. There's one question from Georg Lauer from Harvard for you, Adam. Do we need to think more about boosting the CD4 response together with the CD8 and antibodies? Question mark. The data we saw yesterday from the macaques and also data we have generated in my own lab make me pretty convinced that without good CD4 help, we will not get lasting cure. Unfortunately, they are rarely studied in detail in different treatment trials.
Yeah, all right, thank you. It is an important question because I think Georg pointed out quite clearly that we know a lot about the CD8 T cell response because we've had the reagents to measure it. We know what they look like and we know how they perform. Based on what we know from resolved and acute infection, they're critical, right? And the animal models also say that they're critical for making sure that we can get a good CD8 and a B cell response. And in a similar line, I think it's important that they're part of the analysis of these therapeutic vaccine studies coming down the pipeline because they will induce a CD4 T cell response too, and we have to know how they're responding. So, yeah, I mean, I'm fully in agreement. I think we really need to try and do as good a job as possible to make a well-rounded immune response that incorporates both CD4 CD8 T cell responses.
Harry, may I jump in here? This is Heiner. Can you hear me?
Yes, we can hear you. Go ahead, Heiner.
Okay. Thank you. Adam, following on that one, so great talk, by the way, all talks were really great. Can you comment a little more on the need from your side on the specificity of the CD8s you want to do? We have been discussed, and Georg just asked the question, and his data on POL versus core-specific responses versus S-specific responses, also the Freiburg data. I'm just wondering, when we're talking about therapeutic vaccination, what are we going for? Would it be sufficient to go for core-specific CD8s? Do you want to have a broad one? What is your vision? So should a vaccine include all antigens? Do we need an S-specific response, which is rarely detectable? Can you comment a little more on that one?
Yeah, I mean, it's a great follow-on comment, Heiner, because Robert Thieme outlined that really nicely in his introductory talk yesterday on how those responses evolve with age, right? So the S-specific responses really declined. We've seen it. A lot of people have seen it. So at least, in my personal opinion, focusing on those core and polymerase T cells, potentially even X, which X often gets overlooked because we don't know a whole lot about the T cell responses to X. We don't know how robust they are. But I think we should really focus on these T cell responses we know are still present in the hepatitis B patients. We know that the anti-HBs-specific B cells are present, at least, I think we still need more data on this, but, the papers from Maura and Antonio show that they're still there, and so boosting those has a potential. So at least that's my impression too, that we focus on what we know is present and we try to restore those as much as possible. And we're getting the single cell data on those antigen-specific T cells now. We know we're getting an even better picture of what they look like. And I think rounding that out and getting better data on the CD4 T cells will also be critical to support those vaccine strategies.
Okay. Thank you very much.
But again, one question. So if you would now design a therapeutic vaccine, a T cell vaccine, which antigens to include? All?
No, I would, the core and polymerase for sure would be the main focus.
Yeah.
Okay. All right.
Okay.
Thank you very much, Heiner. We're going to alternate questions from the online platform and the in-person platform, and Carla is up next. So.
Yeah, thank you. Thank you. I really enjoyed these presentations. When we're thinking about a cure, though, I wonder how we should do it as a more personalized approach. I mean, patients respond quite differently, even to the approved therapies with interferon and nucleoside analogs. And so when we're picking these different cure approaches, like, do you have an idea of like where we should be targeting it for, like younger versus older, age-related differences, sex, even genotype-related differences?
Yeah.
Tough question.
That's a good question. So in my view, I think those direct, antiviral agents may have, not that limited to different age groups. Like if you inhibit the virus entry, the replication, all these steps, it should be, works in all ages. And if you combine them together, and that can also, I think, can synergize the effect. So I don't worry about those direct antiviral agents in different age groups, but that's probably Adam's problem on the immunology side. Yeah.
I agree.
You follow up.
Actually, I think Haitao made a good point, because besides interferon, which we know it might work very differently in different patients, but the direct antivirals, depends of the safety, depends of the targeting. Even I think for siRNA depends how well is the targeting of the liver. I think in patients we don't have so many data to really show we have a target engagement in a good proportion of hepatocytes, in some cells, in all cells, and this might have a big consequence also for the to stimulate the immune responses, because if we probably silence everything, the immune response will not know what to catch. But I think it's very unlikely that we actually achieve in vivo. So just to make it short, I think direct antivirals, it's easier to move around with immune modulatory substances, and vaccine is much more challenging. Probably maybe earlier is easier. That's what immunologists say.
Yeah. I mean, I want to touch on something Harry brought up during the public forum earlier, right? So you catch this earlier in these patients, the less likely they are to develop the complications downstream for liver disease, cirrhosis, and hepatocellular carcinoma. Now we got much better data, immune tolerant, or the younger patients, they have immune responses, they're present. If we can target them, we should target as early as we can to try and resolve the infection as early as possible. But right now, it's also clear that these inactive carriers of these e antigen negative patients, it's easier to boost the immune response, and that we might be able to achieve a functional cure in them at a faster rate, and really identify those proofs of concepts, identify those mechanisms we know can eliminate or achieve functional cure that can then be extended to younger age cohorts.
All right. Maybe one more question from the room. Right. Go ahead, Tim.
Thanks. This is Tim Block. So two questions. One, and they're both immunological, but they're for anyone on the panel. One, wouldn't you want to go after T-cell epitopes that are from that aren't normally presented? So if core and pol are what is typically seen, and there are some T cells there, but they're exhausted, wouldn't it make more sense to go after epitopes that might be presented that are not normally presented? That's one thing. So the opposite of what was said in a sense. And then two, I have a question that maybe the panel could help guide the field with. This comes up all the time when we talk about therapeutic vaccines. The good news is that they haven't worked that well, because if they did, that could be so destructive. How do you guide the use of these therapeutic vaccines, assuming they are effective? And you know what I'm talking about, the danger, you must hear it all the time when you propose these, of basically an unbridled immune attack on infected cells. So how can you monitor, how do you balance, how do you monitor the number of infected cells in a person versus a very effective immunotherapeutic? So the first one, would you go after the non-conventional, subdominant, non-conventional epitopes as opposed to the ones you mentioned? And then how do you control for that?
Yeah, I think it's a nice idea, and there are some therapeutic vaccines coming down the pipeline which are actually trying to target those less dominant epitopes. And also there's consideration, just immunologically speaking, the older you become, your thymus involutes, you don't have new T cells to boost, right? So there is a limitation to what you can do in terms of expanding these subdominant epitope T cell responses, potentially. And then you have to know that these subdominant epitopes are presented on the infected hepatocytes, because they may not be as well high affinity for the HLA molecules. There's a number of sort of fundamental immunological techniques that come. As a concept, I think there is real potential, and there's also the potential, say, to target not what are necessarily subdominant, but also immunoproteasome. So there are different ways that a cell will process antigen. There is under normal quiescent conditions, and then there's under inflammatory conditions. And under these inflammatory conditions, they can have a very different profile of epitopes presented. So it's a great idea, and I think it's a potential strategy, but it gets into very granular details of how you manipulate the immune response, which is very, very challenging to actually control. Then I'm totally blanking on the second one. Oh, the uncontrolled immune response. So we've never seen that problem in hepatitis B patients, chronic hepatitis B patients. So the chances you could do it, I think, are very, very slim. Plus, there's now some really nice studies coming out that I think will see that the percentage of infected hepatocytes in liver varies vastly in these different hepatitis B patients. So it's going to be really, really hard to control, because some patients might only have 10% core antigen positive T cells, and some might have 70 or 80, right? So this is something that this comes to be a clinical monitoring and observation, but I don't think that we're going to induce a strong enough immune response to cause fulminant hepatitis with therapeutic vaccine alone.
Okay, Fabien?
Yep, thank you. I had two specific questions. One regarding the decrease in viral antigen levels with siRNA approaches, and whether you think that what is observed in patients is sufficient to reshape the liver microenvironment to make it more prone to stimulation. Because so far we've seen we've seen consistent decrease in HBS levels, but whether this is sufficient to change the liver microenvironment is not known to my knowledge. Do we have any insight on that? And the second question was regarding cccDNA. There's a Japanese groups from Koiki's group have published these two drugs that may restore the SMFAX6 expression. And they claim that it could silence cccDNA, but in our hands, one of the drug didn't work at all. I mean, nitazoxanide, this antiparasitic drug. So I don't know if you had experience, Maura and Haitao, with these drugs. Obviously, the first question was mainly for Adam.
Go first, Adam.
I can.
Yeah, please, because you've done
Maybe,
work on the liver.
yeah, I think I can be shorter, because actually, unfortunately, it is true that some of these substances seems to work well in vitro, but then when you go even in preclinical setting, in animal model in vivo, then you have many more challenges, and you see that they don't work that well. So of course, can be problem of target engagement, whatever, but stability of the drug, many reasons. But then it's where so many drug in the pipeline stop, unfortunately. This is like it is, of course. So yeah, I know we didn't see, but we didn't check so many.
Right. So follow Maura's answer on the siRNA. I think they really needed to improve the delivery efficiency and the intracellular efficiency of those siRNA. So we do a lot of cell culture work. Even if you do siRNA knockdown, normally you cannot achieve a knockout effect. So you still have a residual, the expression there. So I think that also happens in vivo, probably even more complicated. So that's I think one of the limitations of the siRNA.
This is true, but we don't know if this is a real limitation, because if we have some cells that still show something, it might be good for immunological responses, just low level. And that's why I think to which basis whatever, we keep the cccDNA, we avoid new infection. This might also help, just to get responses in some cells, but not risk that we have a real, to go back to the situation at the beginning, right, with real,
Right.
in the follow-up you have a complete rebound, because that's...
Right.
So I'm not sure. I think we need more data on neurological study.
And the siRNA also relies on the host cell risk complex to target the mRNA of the virus. So I think that probably is also the effect is heterogeneous among different cells. So that's my concern. Regarding the second question you have about those two, I can say it's a proof of concept, study. You can target, the DDB1 HBX interaction, or you're targeting the E3 ubiquitin ligase activity that can increase the SMAC5/6, then lead to the inhibition of cccDNA transcription. I think this is a promising, pathway, but it's really, I think the compounds, efficacy and safety, because you basically still targeting host targets, especially the E3 ubiquitin ligase. So I think but that's a proof of concept. I think it can be developed further.
Okay, Adam, did you want to add a few
Okay,
words? Keep it
I'll try
very
and-
short, because we have to close, unfortunately.
So when you talk about the levels of S antigen or levels of HBV antigen, you have to put it into context which part of the immune system you want to talk about. The T cell response or the innate response. T cell response, the effect is much more direct, I think, because you're recognizing the antigen. For the innate response, there's a lot of confusing literature around whether S antigen impairs the innate immune response, inhibits dendritic cells, or inhibits macrophages. This, I think, is a consequence of a lot of different in vitro systems, and I don't think that the data is strong enough to say that S antigen actually impairs the innate response. And you also have to consider what response you want to talk about too, because as Ulla showed yesterday, different cells in the liver express different pattern recognition receptors, which will get targeted by different innate immunomodulators. So if you want to target hepatocytes, using a RIG-I agonist, they're reducing the S antigen might have a more direct effect because the viral replication cycle. But in myeloid cells like monocytes or macrophages, where you're going to, or dendritic cells, where you're going to target with, say, a TLR7 or TLR8 agonist, I think there's less impact the reducing antigen will have with the siRNAs in that case. So I don't think that they'll affect the immune innate response, but they may have an impact on the hepatocyte-specific innate response.
Okay, I apologize to the people online. There are several questions which we unfortunately cannot handle, but some of them will come back in the second discussion and also in the last part with industry. There's one, like a one-line question and a one-line answer, please.
Yeah, very quick question regarding hope that was spoken on from John Davis before. With the very good efficacy of mRNA vaccination seen in the COVID vaccination trials, Adam, maybe for you the question, what do you think are the hopes here for a therapeutic vaccine in the HBV field?
I think that we'll probably see it tested. It would be my guess, but I'm not. I don't know if they're going to be inflammatory enough just on their own, unless they come with an adjuvant that's producing, say, a cytokine that might boost T cell responses. but they've certainly been highly effective in uninfected patients in inducing antibody responses. I think the T-cell response needs a little bit more investigation.
Thank you.
Okay. Thank you very much. So we will reconvene at 10:50 a.m. Toronto time for everyone. So we have a 15-minute break, and we'll hope to see you all back then. Thank you very much.
And let's see now. Trying to share the results of the poll, but the answer is that 57% of the respondees would like the functional cure option, 26 would like the partial cure option, and 17 would like intensified viral suppression in at least 90% of your patients. So that's the outcome of our first round of questions. Thank you.